So let's start. The next point syllabus somatic embryogenesis in unit plant tissue culture. So somatic embryogenesis, that is first of all the embryo production. So embryo production, it is the characteristic features of the flowering plant. As you know, in the after the fertilization, the embryo that will be produced. So this embryo, that is characteristics features of this flowering plant. So in definition of the somatic embryogenesis, that means the formation of the embryo-like structure on the cultured callus tissue is called as somatic embryogenesis. So in somatic embryogenesis, artificially we can make the embryo by using the cultured callus. कैलस फॉर्म टिश्यू कल्चर में आपने देखा है कि जब भी हम एक्सप्लांट का इनोकुलेशन करके हम यदि कंट्रोल कंडीशन में वो ट्यूब रखेंगे, स्लैंट रखेंगे, तब वहाँ पे फर्स्ट स्टेज रहती है कि कैलस डेवलप होता है. तो फ्रॉम दिस कैलस वी कैन directly form the embryos, and this embryo we can use for the formation of the new plantlets. So this process of the formations of this embryo like structure is called as somatic embryogenesis. So here this embryoid embryo like structure on टिश्यू इज कॉल्ड एम्ब्रियोइड. तो हम नेचुरल जो प्रोसेस रहते हैं, नेचुरल प्रोसेस में हम क्या बोलते हैं कि एम्ब्रियो डेवलप होता है. सो हियर इन केस ऑफ द टिश्यू कल्चर द एम्ब्रियोइड इज फॉर्म और एम्ब्रियोइड तैयार किया जाता है फ्रॉम द कैलस टिश्यूज. So there are or the next point is the types of the somatic embryogenesis. So there are types of the somatic embryogenesis in tissue culture: direct somatic embryogenesis or indirect embryogenesis. So first of all, we have discussed about the direct somatic embryogenesis. Somatic embryogenesis, in this method, somatic embryo that are raised directly from the explant tissue without any callus phase formation or callus phase formation. और इट्स एग्जाम्पल है सिट्रस का. तो यहाँ पे हम डायरेक्टली जो एक्सप्लांट है, एक्सप्लांट का डायरेक्टली हम यहाँ पे एम्ब्रियोइड तैयार करेंगे, डायरेक्टली एम्ब्रियो फॉर्म होगा. सो अभी मैंने बताया कि जो फर्स्ट स्टेज क्या रहती है, जब भी हम एक्सप्लांट का इनोकुलेशन करते हैं तो उससे कैलस डेवलप होता है. बट डायरेक्ट एम्ब्रियोजेनेसिस में हम जो एक्सप्लांट है, एक्सप्लांट से डायरेक्टली हम एम्ब्रियोइड और एम्ब्रियो तैयार करेंगे. और यहाँ पे जो कैलस फॉर्मेशन फेज है, कैलस फॉर्मेशन फेज यहाँ पे फॉर्म नहीं होगी. यहाँ पे हम सिट्रस एग्जाम्पल देखेंगे. तो व्हेन दिस सिट्रस, देखो क्या क्या यहाँ पे प्रोसीजर्स है इसके. सो इमैच्योर ग्रीन सिट्रस फ्रूट्स, जो फ्रूट्स हम डायरेक्टली यहाँ पे लेंगे, जो इमैच्योर है, डायरेक्टली ग्रीन कलर के आप देखते हो सिट्रस के फ्रूट्स, तो कैसे आफ्टर मैच्योरेशन उसका येलोइश कलर हो जाता है. तो यहाँ पे हमें इमैच्योर जो फ्रूट्स है, जिसके अंदर एम्ब्रियो मतलब सीड्स जो है अच्छे से डेवलप नहीं हुए होते, जब भी फ्रूट येलोइश होता है तब उसके जो सीड्स, अंदर के जो सीड्स रहते हैं, वो सीड कैसे मैच्योर हो जाते हैं. यदि वो सीड्स हम डायरेक्टली कल्चर के लिए हम क्या करेंगे, जो इमैच्योर सिट्रस के फ्रूट से, इमैच्योर सिट्रस के फ्रूट हम सिलेक्ट करेंगे जो कि ग्रीन कलर में रहते हैं। फिर उसका सरफेस स्टरलाइजेशन करेंगे ड्यू टू द मर्क्यूरी क्लोराइड और डिस्टिल्ड वाटर से हम सरफेस स्टरलाइजेशन करेंगे। देन these surface sterilized fruits we can remove the ovules। जो उसके अंदर जो अनराइप और जो इमेच्योर जो सीड्स रहते दैट इज द ओव्यूल्स रहते, ओव्यूल्स उसे बोलते। तो ओव्यूल्स हम वो रिमूव करेंगे। these ovules we can use for this next procedure। तो जो उसके अंदर जो इमेच्योर ovules हम निकालेंगे। देन these ovules they will be separation of the nucellus by cutting the ovules। ovules को हमें कट करना है, उसके अंदर जो अंदर वाले जो पार्ट है दैट इज न्यूसेलस, न्यूसेलस हमें सेपरेट करना है। एंड these nucellus we can use as a explant and these nucellus we can used for the uh, as a explant and they can use for the next procedure of the tissue culture। so these nucellus we can inoculated on the test tubes which containing the ms media and these forms the somatic embryo। This somatic embryo these are again transferred to the agar medium and after some times they forms the plantlets। so these plantlets then after the formation of the root and shoot these plantlets that will be transferred or they will be hardening at the laboratory first of all, then they will be transferred to the potted plants। this procedure also called as acclimatization or hardening process। so after the plant transfer to the potted plant in or the transfer to the pot, then again this plant will be forms the they will be in pots or in natural condition we can grow this plant so they can grow very fast and they can form the flowers and fruits of this citrus। so here in this citrus plants we can use the immature fruits and from these immature fruits we can remove out the ovules and from that ovules the nucellus part we have directly uh, this nucellus part we have transferred to the ms medium and again after ms medium these parts or these embryos that will be transferred on the agar medium so they can easily form the root and shoot। so here these immature fruit are used in the direct somatic embryogenesis। so these immature green fruits of citrus selected for this somatic embryogenesis, the first procedure हमने देखी कि they will be sterilized with surface sterilizer like the mercury chloride then distilled water or soap solution and for due to the sterilization all contaminants that will be fruits। then the ovaries they will be separated from these fruits with the help of sterile knife। then again these ovaries these will be sterilized with the 0.2% sodium chloride for 15 to 30 minutes। so these ovules that will be or the ovules or the nucellus from these ovules that will be removed with the help of sterile knife or the scalpels। then these nucellus tissues of the ovary that will be transferred into the flask which containing the ms medium or white medium। then this flask or they will be kept for the incubation at 25 to 29 degrees centigrade in the control room, that is the incubation chamber. So here these nucellus that undergo the differentiation and they form the embryo. Then again, the somatic embryo that will be produced in these flask, they will be transferred on the agar medium so they can form the root and shoot. So after the sufficient formation of the shoots and root, they will be transferred or hardening at the laboratory, and then the plant will be transferred on the pots. So here in this way, the from immature ovaries of the citrus fruit, we can got the entire plant. This is the direct somatic embryogenesis. Then, in case of the indirect somatic embryogenesis, production of indirect somatic embryos. Here, example we take as example of the carrot. So here, the carrot seed that will be selected as an explant. These carrot seed, they will be surface sterilized, and then they will be inoculated on the MS medium. Then there will be seedlings of these uh, from the seeds. The seedlings are formed. So these seedlings are the here the cuttings of the petioles will be takes place. These petioles again transferred into the MS medium which contains the 2,4-D. So there will be callus they form. Then again the callus will be transferred in MS medium. Then there will be embryos will be formed, and with the help of this embryo the plantlets are. So here the first step is the select the carrot seeds, and they will be surface sterilized with help of sodium hypochlorite solution for 50 minutes. Then the sterilized, they will be again washed with the distilled water for removing the all traces of the sodium hypochlorite. Then these seeds that will be inoculated with the solid MS medium, and they will be incubated at the incubation chambers. In that, the control condition like the 25 to 29 degree centigrade temperature, then light till 1000 lux, and these all material or test tube that will be kept up to the 16 to 24 hours. So these seeds they will be started germination uh, up to the 2 to 4 weeks. Then, after the formation of the seedlings, the young and small petiole of the germinated seedlings, they will be removed by sterile knife, and these transferred on the MS medium, which containing the 2,4-D in 0.45 million. So here again this culture will be incubated in the incubation chamber. Then after the formation of callus, these callus they will be subcultured on the fresh nutrient medium which contain the amount of the 2,4-D. This callus tissue they will be produces the or they shows the small embryos, and these embryonic initials which develops into the plantlet. So uh, in this indirect process, the time as well as the nutrient medium required in the high amount. So the plantlets, after the formation of plantlets, they will be transferred into the greenhouse uh, for the proper growth of this point. And after proper growth of these plantlets, these will be transferred on the main field. So here in indirect process of the somatic embryogenesis, it is quite a longer process, and here the reculturing of this material as well as the different nutrient medium they will be required. So commercial aspects of the somatic embryogenesis are the from small tissue we can got the many plants, or we can got the number of plantlets from the small tissue of this plant. Then, if the plants are showing all superior characters, uh, or the plants they show the superior characters as that of the parent plant or mother plant. So, when the selected plant for the tissue culture will be superior, ultimately जो हम plantlets सब मिलेंगे, that will be also a good quality or they will be superior. Then those plant will be raised from the somatic embryogenesis. All plants are identical and they are not showing the variations. Then any somatic cells they are develops embryoids or embryo every time, which is rare as compared to the somatic cells. So these plants raised from these somatic embryogenesis, they are disease free or disease resistance. They have embryonic viability or the embryonic viability that will be increases. Sometimes what's happened in some plants, all embryos that are that cannot be grow. So like that plants, if we can use the somatic embryogenesis process, we can easily uh, grow these plants on the tissue culture and you can got the number of plantlets. So in case of the citrus, generally citrus all seeds are not grow. यदि हम जो लेमन के जो भी सीड्स यदि वो नेचुरल कंडीशन में हम पॉट में ग्रो करेंगे तो वहाँ पे ग्रो नहीं होते. यदि हम सिट्रस का यदि थ्रू उसका जो से न्यूक्लियस कल्चर थ्रू वो ग्रो करेंगे तो इजीली वहाँ पे ऑल एम्ब्रियोज ग्रो कर सकते. सो दीज आर द बेनी importance or commercial aspects of the somatic embryogenesis. So here the somatic embryogenesis will be over and somatic embryogenesis. So the somatic embryogenesis, lastly I should revisic embryogenesis, that means it is the formation of the embryo from the or embryo like structure from this cultural or from the callus tissues of this. So these somatic embryogenesis, they are differentiated into two types: direct somatic embryogenesis and indirect somatic embryogenesis. डायरेक्ट सोमेटिक एम्ब्रियोजेनेसिस में हमने सिट्रस का एग्जांपल लिया. सिट्रस में हम क्या करते हैं कि जो इमेच्योर फ्रूट्स हैं, immature fruits, we can remove the ovaries, and again in the ovary we can remove the nucellus of that ovary, and this nucellus we can grow on the MS medium. So these nucellus they can form the embryo, and this embryo that will be producing the plantlets. In indirect somatic embryogenesis, we take as example of the carrot seeds. So here carrot seeds, first of all, we have sterilized it and it grows on the MS medium. Then the if the, after incubation the seedlings are formed. So these seedlings, the petiole that will be again transferred into the MS medium. This petiole they forms the callus, and this callus they will be again transferred or recultured on the new MS medium, which is high contents of the 2,4-D, and this callus they form the somatic embryos, and from the somatic embryos again the plantlets are formed. So in this way, directly as well as indirectly, we can got the somatic embryos with the help of tissue culture. So there are some commercial aspects of the somatic embryogenesis. This technique is used in case of the many plant species. So if we use this technique, we can got the disease resistance as well as superior disease or high viability plant. And this plant from this using this technique of somatic embryogenesis, all the plants are identical. There is no variation. The plants will be superior as like the parent plant. So here the somatic embryogenesis point is over.